Okay, so we should be recording now. Now this, yes, I'm in here twice at the moment. I should. Problem that we've got the audio twice. Okay, that's weird. Okay, can I adjust the audio at all? No, I don't think so. Let's see if I can adjust the audio at all. More. Yeah, we've also got construction going on just outside the door, which isn't very helpful, I'm afraid. Okay, so I'll try and speak a bit louder. Is that better now? Can you hear me okay now? Yeah, unfortunately, I can't. It's the setup in the lab. I can't change the microphone or anything else. I have to use the setup they have here. Okay, now. So as I mentioned in the message, you need to be running JMP. So for those of us in the classroom, you should be able to find JMP icon on the desktop. So I'm going to switch, so I can get Zoom back. I'm going to switch Zoom to sharing the screen. Okay, can everybody see my screen okay? So for those of you who have installed JMP at home on your laptop, I hope you have downloaded the trial. Now, I will try and get JMP to give us an extra license. The trial license only lasts for 30 days, and unfortunately, the class lasts for more than 30 days. Okay. So I don't need you guys who are remote to turn on your video, but I do need you to show me that you have got JMP running okay now. Let me open. I want to check who I have here. Okay, so let's just see. First of all, I can see in Zoom who is connected. So I have Stephen Leung. That's Miss Leung Yi Man, who's present. I have Fan Min, present. I have Francesco Recchia, present. I have Matthew Fung. I have Jackson Lau. I have Kenneth Chung and Nick Zhao. And who are you two guys? Xi Zhao. Oh, Gao Ji Zhao. Okay. And Zhang Jiaxiu. Okay. So we don't seem to have Rajesh, and we don't seem to have Edmund. So we have 9 people. We have, and I'm double counted on here. So we have 7 online and 2 in the classroom. Okay, so have you guys both got the JMP loaded correctly? Okay. So what about those of you who are in Zoom? Please indicate to me in chat. You can send a message directly to me whether you have any problems with JMP. Okay, everybody got it okay? The trial version is fine. So I'll try and get hold of JMP. Previously promised me that they would let me have access to licenses, short-term licenses, to cope with the trial problem when I explain the difficulties. In fact, if you find JMP helpful, the on-the-hub price is very reasonable. Just $30 US for 12 months at the moment. Now, on the hub was until 2 months ago selling JMP version 14. 2 months ago, they switched to 15, and then this week they suddenly offered me a free upgrade to 16. In the lab, we have JMP Pro 15. So the trial, I'm assuming if you just downloaded it, you have 16, correct? Yeah. Now the differences in the different versions of JMP are usually very small. They just generally add things in the more sophisticated part, which won't be our concern. So the primary issue is actually the differences between Mac and Windows are generally greater than the difference. Is that? Let me just check if I move the mic. If that helps a bit. Is that better now? Can you hear me better now? Or just the same? Okay. Now, in terms of materials, so let me just check. So if you go to Moodle, some problem with the mic. Okay. So if you go to Moodle, let me go back to Hong Kong U portal. I have to log in again. Yeah. So let me do this on the PC. Now I'm going to work out which browser is running here. Okay, we have it. Let's have another tab here, Hong Kong U portal. I'm going to log into my account. Okay, so you won't see quite the same view as I get in the portal. So the crucial issue is that there is a courseware folder in Google Drive, which you can then click on the shared folder. Oh. So then I've got to log in yet again. So you need to use your Iqrm Connect account. You can't use your normal Google account. Okay, so at the moment there's nothing there. So I'm going to put some files in there for us to review. So let me just do that. So for now, I'm going to put 2 files in here. I will put in some data sets as well later, but I want to check that those data sets work okay in the latest version of JMP. So there are 2 files here now. So you will need both of these files. You don't need to necessarily download it immediately now. I'm going to show you about the content of these two files. So these two PDFs contain the materials for this course. Let me see if I can download it on this computer, which doesn't belong to me. Yep. So the DA book is the one that you need to download and open right now in order to follow along. Now, this is not showing up in Zoom, so I need to move it. So I assume you can see it now, correct? I put it in my Zoom window. So this course book is Creative Commons licensed. That means basically you can share it with anyone you like. This course book doesn't have a lot of information, so probably nobody who doesn't take this course is going to find of interest. Now, the other book, the other PDF file, has a lot more information, and I'm going to talk about that in a few minutes. But this one, I need to go through it a bit right now because it talks about what we're going to do in this course. Okay? So, first of all, the course aim. It says, I aim to equip you with computer-aided skills in organizing, transforming, and analyzing data. It emphasizes proper usage of data analysis and intelligent interpretation of results through hands-on learning during and after classes. And so we're going to use JMP in the course. Now, when it comes to the assignment, I don't require you to use JMP, and I'll say something about that in a minute. But we're going to use JMP in the class so that we can all follow along together. Now, JMP is not free, but it is, in my view, still the most user-friendly of all the statistical packages. It's not the most powerful. So I have no difficulty with the idea that people prefer other things, and it's not free. So you can, of course, choose to use R. And in fact, I will be running a workshop on R for the grad school next week. But R is much harder to get started and is not nearly as user-friendly. And it's much harder to get the graphics to work in R, whereas in JMP, the graphics are fully integrated. So it's much more user-friendly, in my view, than something like SPSS. And it's very flexible. So the learning objective is to develop practical skills to undertake meaningful statistical analysis of data sets. It says to fit linear models and undertake diagnostic checks. We'll go beyond simple linear models, but we're going to look at linear models in the broadest sense. Okay, so the assumed background. So this is an important issue. What do I assume you already know? So I assume that you already know how to do basic data display for a single variable, estimation, hypothesis testing for means and proportions, goodness of fit for 1- and 2-way tables, the basics of probability and linear models. So this is chapters 1 to 10 in the other book on that website. So I expect you to review those chapters in order to make sure your understanding is in good shape. So the only exception is chapter 4. Chapter 4 is a simple introduction to qualitative analysis. Obviously, you don't need to understand that for this course, but I encourage you to read that chapter just to increase your knowledge. So for this course, we're going to cover chapters 11 to 16 of the IQRM book to cover modeling, and 17 to 19 to cover JMP in practice. Okay, so we're going to switch to that book in a few minutes, but I want to say a bit more first. So you can't see the PDF. The PDF should have shown up in Google Drive, as I showed inside Moodle. Oh, sorry, I switched screens, didn't I? Let me just go back. So inside the Google Drive folder in Moodle, you should be able to see those two files, okay? Okay, so I've got to find my way around this keyboard. Yeah, there's page down. So this course is going to cover summarizing data. Again, I assume you know this, but we're going to go through this using JMP just to see that we know how to do it. We're going to look at associations. We're going to look at hypothesis testing, analysis of variance, regression models, basics of time series. We're going to look at a set of nonlinear models. We're primarily going to look at what's called generalized linear models. And we will say something briefly about multivariate data analysis, although we're going to be fairly simple in what we do. Now, the important issue is assessment. So it's a pass-fail assessment for this course, which is based on a report that you're going to submit. So let me say something about that assessment. Make sure you understand. So I want you to investigate a data set and write a report on your findings. So the deadline is 4 weeks after the end of the final class. You're welcome to submit it sooner. If you need more time, you need to email me with a proposed submission date, okay? And the simple requirements are that the data set should have at least 50 rows, at least 50 cases. There should be a continuous dependent variable. Why continuous? Basically because that's the primary situation that we're going to look at in this course. So I'm willing to discuss that for people who have a data set they want to analyze that's not a continuous dependent variable. But in general, that makes life a lot simpler for the tools that we're going to look at in the course. And then I want, amongst the independent variables, at least one covariate and at least one factor. So there can be more than one covariate and more than one factor, but I want at least one of each type to make sure you've understood both the regression models, the analysis of variance models, the analysis of covariance models, all of these, that you can understand the basics of the analysis. You can always choose a subset of a larger data set. So I encourage you to find a data set that's from your discipline or another data set for where you at least understand the research question, right? So there needs to be at least an implicit research question to investigate, even if the research question is just what's the best model for this data set, okay? But I want that you understand what the variables are. I don't like people analyzing simulated data sets, which I consider in general unrealistic, okay? Now, I've given you a couple of links if you want to go and find data sets. So Kaggle, for example, you have to sign up if you want to use Kaggle. In Kaggle, some of the data sets are much too big, but you could always choose one of those data sets and simplify it down. And then statsci.org also has some data sets. But again, if you want to use a data set that you're using in your thesis or one that you've got from colleagues, that's fine with me. It's not a problem. If you can't find a suitable data set that you can use for this course, you're welcome to go and find anything that's of interest. So if you're interested in sport, for example, you could choose a sports data set, right? Or if you're interested in culture, you could find a cultural data set. I don't really mind. As I said, what I care about is that you understand the context of that data set and you understand what is a meaningful model in that particular context. Now, with that data set, I want you to, oh, go away, Microsoft. Within that data set, I want you to write a report. So it says the report should not exceed 6 pages of A4. You can also have up to another 6 pages of appendices. In other words, things to refer to, but which I shouldn't have to read, in a single document with your name and student number inside the report. And that report should be submitted by email to me as a PDF file. So I don't use Moodle for this situation because the deadline is not a very fixed deadline, and I want to make sure I don't miss it. Now, you also, in addition to the report, need to attach the data set. And the three possible formats would be Excel, JMP, or SPSS. In fact, I would accept a CSV file as long as you tell me how it's organized. Now, if I consider your report inadequate, I will return it with suggestions for improvement. So most people in the past did not manage to submit a report that was fully adequate in the first round. So that's a normal process. I expect it to be iterative. And then I've given you some ideas about how to write this report. Again, it's not very long. It should be written in the third person. It's not I did this or we did this, right? Reports are usually written in sections, and the sections I expect to find are a title page, which should have your name and student number and the date it was written or submitted, a methodology of procedures where you outline how you did the analysis, how you investigated the area, a brief introduction, so that would be explaining the context of the data set and the analysis that you're trying. And then the findings, analysis, and results are the main body of the report. And I said you must, as a minimum, include discussion of the best model that you found for your data, an explanation as to why you think that model is the best one, a summary of the diagnostics you have examined for that best model, and a summary of what other models you examined, okay? And then a conclusion should be drawn from that analysis that's clear and concise, should link back. And in the conclusion, you shouldn't be introducing new information. It should just be covering what you have deduced based and shown in the findings, analysis, and results. And any limitations should be referred to here. So it may be that every model that you fit has some weaknesses, then you at least should make some reference. So we have some idea how much we can trust those findings. And then a brief bibliography, that would normally mean a reference to where the data set came from and any other specific materials which are relevant. And the appendices, you can include detailed statistical tables here if you think they might be needed for reference. Okay, so the context for this, so this is what we're going to talk about is the key elements of quantitative methods, which is what we're looking at here. We start out with counting. This is the most basic level, things such as how many boys in our sample. We then move on to testing hypotheses, such as, for example, are there more boys than girls in a population? We then move on to modeling variability. So this is essentially what we're focusing on primarily in this course, which is, for example, how much do age and gender determine academic results? We can also look at prediction. That's not a focus in this course. So prediction is the hardest thing. That would mean something such as, can I predict whether a boy age 16 from a band 1 school will pass an exam? That's obviously much more difficult because that involves individual variation. Okay, so in our research analysis process, we're going to start out with what are the research questions and what hypotheses do we want to investigate. Secondly, design and implementing an experiment or other data generation process. That could be a survey that is capable of answering the questions. So we're assuming here that A and B have already been done, right? We're not in this course expecting you to go out and do those. I'm assuming that the data set should already implicitly tell you how the data was generated and what the key research questions are. So what we're going to focus on are the next things, such as what are the dependent variables? And we're going to assume in most of the examples here that it's a single dependent variable. We will look a little bit at multiple dependent variables and multivariate analysis, but primarily we're going to look at cases with one dependent variable. You need to understand the type of variable, whether it's nominal, ordinal, or interval scale. Again, there is references to that in the book, including any constraints on the dependent variables, for example, that they must be positive. We're going to identify all the possible independent variables and their type. We're going to construct a model that explains how the independent variables may affect the dependent variable, that includes all the possible models under the hypothesis of interest, which means that we aim to identify a plausible process that generates the dependent variables given the values of the independent variables. We're going to then use statistical methods to identify a good model. So a good model means generally that it fits well, is relatively simple, that we can interpret, and which the diagnostics confirm is reasonable. And then lastly, identify the research questions and accept or reject the hypothesis. Okay? Now, in this course, we're going to do this on a problem-based learning context, which means we're generally going to look at data sets and start from there. We're not going to start with the theory. I'm assuming that you know the basic theory, although we can talk about that in some cases. So we're primarily going to look at data sets and ask the questions, what can we learn from this data set? What is a reasonable model? Can we confirm whether this model actually does seem appropriate? So the three key dimensions are, first of all, are we using methods that are appropriate given how the data were collected and what type of data we have? Secondly, how do we interpret the results? And then thirdly, diagnostics. How can we check whether the methods are appropriate? If they're not appropriate, we want to try and find a better method, okay? So using computer packages makes diagnostics relatively easy to use and allows us to try alternative methods and see if this affects our conclusions. So this is sometimes called sensitivity analysis, right? Okay, so that's basically all we're going to look at in this document here, except what I have remained here is a list of data sets that we're going to look at. So the first, I think, data sets 1 to 15 are included inside JMP, right? So these ones, so at least for the first two weeks, at least this week and next week, all the data sets we look at will be using the sample data sets that come with JMP. And then in the later 2 weeks, we're going to look at some additional data sets. So I will put these in the Google Drive folder. So I think it's data sets 16 to, how far do I go here? 26. Yeah, so 16 to 26, I will be placing in the Google Drive folder for us to look at later. Okay? So we're going to go back now to the other book. So this is the IQRM book. So this book is the book I use for all my courses and workshops for the grad school. So this is also Creative Commons licensed. So you're welcome to keep this book forever. You can share it with anyone you like. So those of you who've taken my courses before will know this particular Creative Commons license means you can use the materials, but you need to give me credit. You cannot sell it. You can give it away, but you can't sell it. And if you share with other people, you should share in the same way. Okay, so this has been updated this week. I've updated in the light of one of my workshops. So we're not going to go through these earlier chapters, but I encourage you to review them and make sure you understand all of these things. So there's an introduction to research methods, discussion of association and causation, the fundamentals of research designs, including representativeness and sampling and non-sampling errors, the basics of qualitative research, which I believe all researchers should know the basics of, so that if you read a paper and that person makes some reference to qualitative methods, you have some idea what they're talking about. Measuring instruments. So measuring instrument here is an abstract notion. It could be a piece of physical equipment in a laboratory, but it could also be a questionnaire, or it could be an assessment tool that you're using on some students. These are all different forms of measuring instruments. So we talk about the criteria for a good measuring instrument: reliability, validity, and precision. And importantly for this course, we talk about scales of measurement. So nominal scale means there is no ordering of the categories. Ordinal scales means that the categories are ordered. Interval scale means that differences in values are meaningful. And ratio means that percentages of values are meaningful. So if you don't have these parts clear in your head, you may need to revise this chapter. Chapter 6 on probability is not very important for this course, but if you don't have this part clear, you may want to look at this. The statistical computing is just basically one page, the things that I've just talked about just now. And then we come on to the reality of doing statistics, summarizing data, which we're going to start with today, and then estimation, hypothesis testing, relationships between pairs of variables, which we'll also move on to later today, and then coming on to the more crucial parts of this course: multiple regression, analysis of variance, general linear model, generalized linear model. Experimental design, we will not really talk about too much. Time series, we'll look at some simple examples, okay? So we're going to go to chapter 17, first of all, which is about the basics of using JMP. So let me, now, can I download this to somewhere? I'm just going to have to switch back and forth, I guess. Okay, so that means I have to actually scroll this down. Get 214. All right. It scrolls pretty quick. Okay, so chapter 17 is the basics of JMP. So let me pull up JMP. So I'm going to refer primarily to JMP rather than to what's written in the course book. So I encourage you to keep that open so that you can refer to it. So JMP, the key concept is a table. So we can create a table in many different ways. So a data table in JMP is essentially a spreadsheet table. So you can think of it just like an Excel table. The issue is that in general, we're going to assume that one row is one case and one column is one variable, right? So that's generally works very simply. The main variation is that we may use one column as a count or frequency column. So if we look at, so let's create a new table. So file, new data table. So it starts out with one column and no cases. So let's say I'm doing a very simple 2x2 table. So if I click on this first column, if I double click on this, it lets me change the name so I can have gender. Now, JMP asks basically what's the data type and what's the modeling type. So data type means, is this numbers or text? So I'm going to input the gender as text, male and female. So I need to click on the data type and change this to character. That means that it will allow me to put text in this variable. And then you see it automatically changed the modeling type to nominal, meaning if it's text, it assumes that usually it's nominal scale. Now, we can change it to ordinal scale, but then we would need to tell it what that ordering is, right? So if we have males and females, and in some case, in this context, hopefully not, though we wanted to say that males were and females had an ordering, We could do it, but we would need to tell JMP what the ordering is. It can't work it out for itself. So I'm going to leave this, of course, as nominal. We can just click OK. So you'll see this is now changed to gender. And if you look on the left-hand side, you will see this little icon next to the name of the variable. This icon means that this is nominal scale, right? It means there's no ordering here. So what might my second column be? So I want to add another column. I can choose new columns, or I can double click on the second column. So you can't see that perhaps very clearly, but if you double click in this place here next to gender, you can double click again, and I can now put something else in here. So what might I put in here? What should we have? Gender by faculty. No, I won't use faculty. What shall I use? Gender and age group. Let's put age group. So age group, again, I want to be able to put in here, perhaps numeric. I'm going to put the starting of the age group as the age group, or I can number them. But these I'm not going to put as continuous, because continuous means that the values I input should cover all the possible values. So I'm going to change this to ordinal, because my age groups at the very least should be on an ordinal scale. Right? So if I click OK, now I don't have any data yet, but I can again just double click in here and just type a capital M in here, or I can actually write male if I want. And you'll see now I have one row, and you'll notice it's put a little dot for age group. That means this data is missing. I haven't yet told it what the age group is. And then perhaps I have a second male group, second male, and then a female. So I can just type these in. And then for the age group, I'm going to have age group 1, age group 2, age group 1, and age group 2. So in this situation, in fact, I can even have a third. Maybe I'll make this a 2 by 3 table. I'm perhaps also going to have an age group 3. So I'm going to put female 3. So I now have 3 age groups, but you'll notice that the icon for age group here, this icon here means this is ordinal scale. It's not continuous. So it's put the sort of increasing bars to indicate this is ordinal scale. In fact, if I also had another column, so I'm going to now add in a column for counts or frequencies, right? I can call this either count or frequency. So let's call this one frequency. So this one I do want to be numeric, and I will leave it as continuous, because this is actually a count. How many times did this combination of gender and age group occur? So I might have 10, 20, 30, 20, 10, 20, right? So I've written down, so rather than having to put in 10 rows, each of which have gender male and age group 1, I can put in the counts in another column. Now, again, JMP will not automatically know this. I will have to tell it if I want to actually use this column as a frequency variable. Go away. Okay, now the age group, as I said, this is ordinal. So by telling it's ordinal, it will not assume that the difference between 1 and 2 is the same as the difference between 2 and 3. Right? If I put continuous, it will assume that those differences have the same meaning. Ordinal scale means just that 2 is greater than 1 and 3 is greater than 2. Okay, and I could change these to being labels, or I can add labels in. So if we look at the columns, and if I look at the column information, then for age group, I can click on the column properties, and somewhere down here, if I can find it. I've got value labels. So I can now tell JMP age group 1, this value actually is maybe 20 to 29. That's the age range. And then value 2 perhaps is 30 to 39. And 3 is 40 to 49. Okay, so once I put this in here, if I come back here, you'll see now it is showing me the labels for the age group, right? So I don't have to keep typing in 20 to 29. I can still type in 1, 2, 3, but in all the output, it will show me the values for that. Okay, now, because I have told JMP which variables are nominal, ordinal, and continuous, that means it can be a bit smarter. And so if I tell it I want to analyze fit y by x, in other words, I want to see the relationship between gender and age group. Then if I do this and I say I want to know how age group depends on gender, right? I can either drag gender and age group into these places, or I can click on it. Now, the thing that I haven't yet done is frequency. So if I click on frequency, I can drag that into the frequency box. That means I'm telling JMP I want to look at the relationship between age group and gender using this frequency column to tell me how many times each of these values occur. Okay? And if I now click on OK, you'll see it gives me a plot. So this is what's called a mosaic plot. And this mosaic plot then illustrates what the table tells me. So tables, unless you're somebody whose brain is wired in a way to understand numbers implicitly, it's sometimes hard to work out what it's showing you. So here we can see that there is a difference, right? You can see that the proportions of males and females who are aged 40 to 49 is similar, but the proportions who are in the 20 to 29 or 30 to 39 are a bit different. And indeed, it's telling us down below that the association is significant at about 6%. So this is not quite statistically significant. I just made up these numbers from my head. I wasn't even thinking about what I was typing in. Right, but we can immediately see we've got the table of counts. By default, it gives us all the different percentages. And by default, it gives us both the G-squared likelihood ratio test and the Pearson X-squared test for association between age group and gender. Right, that's what it does by default here. Now, in fact, given that age group is ordinal, we maybe want to do a different test. Right, so there are other tests that we can do, but we're not going to worry about that right now. Okay, so in this case, you'll see because it knew one of these variables was nominal scale and one was ordinal, it already had some idea of how to summarize the results for us and some idea of what tests to apply. Okay, so that's very easy if we're creating our own data file. What if we are inputting it? Let me see, what was my next point? So we've talked about entering data directly. We can import data. So for example, now let's have a look. So I don't know where anything is organized on this particular computer. Let me see if I can find. Ah, here we go. So on the computers in the laboratory, you should be able to see a link to JMP sample, right? And then inside that, it has a data folder. This is the data sets we're going to look at generally. There is another folder called import data. Oh, somebody's emptied this. Oh, no, they haven't entered it because it's showing me only the JMP files, whereas I want to see all files. Okay. So if we change that filter, we can see all the files in the import folder. So this animals.txt one, we can try opening this. So if this file is already in Excel, then usually it assumes that the Excel file has that the first row is the variable names, right? And the rest is data. But if it's a text file, in general, JMP doesn't know what the format is going to be of the data. So you'll notice here it says, let me turn off the use default program. It then allows us to open that file in 4 different ways. As data, using the preferences, meaning it should already know what to do with it. That's not generally a good idea. Best guess, again, this is not a good idea for a text file. So generally for a text file, you should always be using preview. That means it will show you what the current options are so you can see whether it's set correctly for this particular text file. So we're going to do data using preview. We're going to open it. And so this particular file, you will see that it does not have the names of the variables in the first row. This particular file starts with data in the first row. Whereas if you created it by exporting from a different statistics package or from Excel, usually you would have put the variables in the first row. So we're going to have to work out what the variables are for ourselves. Now, because the first column has text all the way through and the fourth column has text all the way through, it can work that out for itself. It is guessed that these are delimited fields. Delimited means there's something separating the columns. And it's worked out that there are tabs between them. So in a CSV file, that means comma separated. So if this was a CSV file, we would need to select comma as recording end of field. Right? So if I put comma here only, then it doesn't recognize these as separate. We can put different options in here, and the preview lets us check. And you'll see this option says, file contains column names on line, usually on 1, right? If I put this, then it would think that Fox was actually the name of the first column, and 4 was the name of that column, which is clearly wrong. Right? So we need to look at this a bit more carefully, and then also end of line. So depending where this file came from, whether it comes from Linux or Unix or Mac or PC, the end of a line can be a carriage return, a line feed, or both. So the default is it puts all of these. So basically any of these, by default, it will assume is the end of a line, and we're going to a new record. Okay? Now, we can also sometimes have data which is in fixed width fields. So that would mean that there are a fixed widths. So maybe the first column is always 3, and then the second and third are both always 2. In fact, that's clearly not right for this one, right? So if we did this, we would clearly get a mess. It allows us to create column dividers to work it out for itself. But clearly, there's no easy way to do this. This cannot be correct for this data file. Okay? So once we think it's all correct, we click on Next. It's now going to allow us to create column names. So clearly, it's just put 0, 1, 0, 2, 0, 3, 0, 4, because it has no idea what these are. So this is clearly a species of animal. We don't actually know what these are. This one is clearly season. Right now, as it turns out, this one is actually distance. This is the distance that these animals travel. And this one actually is the individual. I know this already, but you would need to go and look up and find out what do these columns actually mean. Right, so if I put these in, I can then click import, and then I now have my JMP data file. And you'll notice for species, it correctly identified this as nominal scale because it's text. And similarly for season. Individual and distance, it is put as continuous. So for distance, that's okay. Actually, it's clearly been rounded or rounded up or down or in some way to the nearest whole number. But clearly the individual should not be continuous, right? The individual actually should be nominal. This is actually the individual within species. So the individual I want to change. If I click on this, you'll see I can change it. So I actually want to change this to nominal scale because JMP wasn't smart enough in this case because it had no way to know that this was really nominal scale. And you'll see that the icon now changes. Okay, so JMP is fairly smart, but there are situations where it cannot work things out for itself, and you need to use your idea, right? Yeah, I am using a JMP sample, but these are the sample imports, right? So this is the sample import data. Okay, so it's not hard to import a text one. Text is probably the hardest one to do. So let's look at, we can also look at importing Excel. I think there is, well, let's try, we can try, yeah, there's one here called fill weights. So the only real issue here you'll see is whether the row 1 are labels or not. Right? So we'll just try best guess because usually for Excel it can work out. So let's have a look to see how that works. So you can see actually there is a problem here with this one because the headers are more than one row. Right, the headers here are 2 rows. So there are actually 2 rows with the column headers. The data actually starts in row 3. Okay, in fact, this one is even more messy because this one does not have the same number of rows in the different columns. Right, but this one at least we can sort of work it out. But you'll notice that this one is a bit problematic. In fact, I still got it wrong. Oh, it was actually more, it was three rows, I think, contained the data. So I got that one wrong. Let me try it again. So it usually will be obvious. Where were we? Yeah, fill weights. So this is a messy one. Yeah, so actually it's 4 rows. Yeah, 4 rows contains the column headers. So then it will accurately put missing values in for the other values where there is no value in that particular row. Okay, so it's not too bad, but in fact, the way this is set up, it's messy. We don't really want the first row. We could have actually thrown away the first row. It just says this column contains blah, blah, blah. So it's basically a note. It's not really the data. So let's look at a more reasonable Excel one. they've changed the samples from last time I looked at this. What about this one? Restaurant tips. Let's see whether this one looks more reasonable. This one looks more reasonable, but it's the first column. It's only the first column that contains the column headers. So data starts on row 2. And this one, it looks like it's pretty good. It's able to determine generally some of these are text and some of them are numbers. So again, the main issue is you need to check carefully whether the number ones are really continuous, nominal, or ordinal scale. Okay? So importing is generally not too hard. And in fact, JMP is pretty smart. If I remember correctly, I don't think there is a sample here. Yeah. In fact, if I remember correctly, for SAS files, it can normally read them directly and extract most of the information. It's pretty good at importing different files. Okay, now, of course, there are even other options as well. We can, for example, even generate a random file. I'm not going to bother to keep that one I just generated. We could even create a random data table. So for example, this might be income. Let's say this data, this is income data. And in fact, we want to do it randomly. How do we do this? Well, actually, it turns out this is moving ahead a little bit into a formula. So one of the things that's nice about JMP is that it generally encourages you to write down a formula for the different columns. So, for example, I might, if I want to generate income, it's a little hard to put in a simple formula to generate income. So what I'm going to do is generate a formula for log income first, and then from that, I will calculate the income. And I'm going to do it log to the base 10, because I prefer log to the base 10, because I can do the conversion in my head. Okay? So my log to the base 10 income, I'm going to assume I'm going to put a little formula in here. So what's my formula going to be? So random. I'm going to write it, assume it's random normal distribution. And I can say what is the mean and the variance of that normal distribution. So if I want the, so what's the average income? Assume the average income is $10,000. So 10,000 log to the base 10 of 10,000 is, so 10,000 is 10 to the power of 4. So if I put a mean in here of 4, I'm not going to bother about the variance for the moment. Now, at the moment, I don't have any rows. So I can add rows. I can say I want to generate 500. I add 500 rows. So you'll see now I have generated 500 values with a mean of 5 and a variance of 1. So we can actually check that. So let's illustrate quickly the distribution for that log income, just to check we've done what we thought we did. This tells us that the mean is 4. Sorry, I said 4 or 5? I said 4, didn't I? Yes. 4 is 10,000. The standard deviation is 0.95, right? So it's approximately a standard deviation of 1. So this is a random sample where the mean of the distribution I sampled from was 4, and the standard deviation of the distribution I sampled from was 1. Right, so we get pretty close to 4 and 1 when we look at the summary statistics. And now I want to convert that into a log, into an income variable. So I create a new column, income. And this one initially is missing because I haven't told it how to find it. I can again put a formula by right-clicking on the column. And here, what is my formula? So income is just log income exponentiated. So I start out with log income. And then transcendental, we're going to look back at these later. So actually what I want is 10 to that power. So in fact, I shouldn't have done this, right? Because by using log base 10, I have a slight problem. But what it has in here is exponential. It doesn't have 10 to the power. Where's 10 to the power? 10 to the power. I've forgotten where 10 to the power is in here. Okay, let's put exponential. And then we need to multiply this by logs. What have I got? I've got to work this out again, right? This is wrong because I want 10 to this power. Let me just check. I think I can do it directly. Let me try again. I want to start with log-based income. I can choose up here in the formula power. I drag this up to the power part, and I want 10 to be the thing that's to that power. Okay, so this will be 10 to the power of log 10 income. Let's apply that, see whether that looks right. So if I now look at the distribution of this, so you will see that actually the mean, oh, what have I done wrong? The median is indeed roughly 10,000, but the mean is much bigger. Why? Right, because this is on a log scale. So +1 and -1 have very different meanings when we look back on that scale. So in other words, we've got people with 10 times the income and people with 10 times less income. So we're saying equal proportion of getting $1,000 and $100,000. So the median is about right, but the mean will be very different. Okay? But we can simulate all of these. We can simulate a data set. We can then use a formula to convert it to a different scale. So it's very easy then to do to generate our own data set that's simulated. What haven't we done? So let's have a look now about one of the nice things about JMP, which is better than most of its competitors, is the ease of selecting things in different ways. So for example, if we looked at this distribution here of income, you will see there are some very large values here on the income scale. So this dot here, for example, there's one person who's got an income of 600,000. So what if I wanted to highlight that individual? I want to find which individual that is. So you'll see the 3 dots on this window. If you put your mouse over the 3 dots, the menu comes down. Okay? Now, what we want to do is, I don't know whether you can see on my screen, but on your screen, you should be able to see there is a lasso. If I put my mouse over it, it actually says lasso. All right, so what's a lasso? So lasso is like cowboys used to use. If you've ever watched a Western, the cowboy is riding along on his horse and he has a rope. And he basically has a loop on that rope, and he's skillful enough to wind the rope around his head, throw the rope out. The loop is supposed to go over the head of the cow that he's chasing, and then he can pull on the rope, and the rope tightens around the neck of the cow. So basically, lasso is you do a circle around something, a very rough circle, in order to select. So if I click on the Lasso tool, you will see my icon has changed now. Instead of having a pointer, I have a lasso. So if I now click and drag, I can draw a very crude circle around that point. And it has now selected that particular data point. If I come back to my data set here, you will see I have selected row, in my case, 185. Yours may be different because this is a random data set. But whichever one you've selected, it will show as highlighted back in the JMP data table. So this is very nice. So selection of a row or a set of rows works across all the windows in JMP. So I showed you how to select using Lasso. And once we have selected a row, the rows tools allow us to do various different things. What can we do? We can color it. So we could make that one a nice bright blue, well, I use a dark blue. So then if I come back to my other window, you will see now my data point has changed color to blue. I can also change the marker if I like. Instead of being a round dot, I can make it a star. And you'll see it's changed to a star in the window. So it's quite nice. I can use different ways. So I can, for example, use different colors or markers for gender or age group or something else, or for the ones which I find interesting. And it works across all my windows instantaneously. All right, so I can also select in other ways. So instead of selecting in that way from the graphic, I could come here to rows and say row selection. So if I come to row selection, I can do the most flexible one is select where. So if I choose select where, I can put conditions on my columns to select. So for example, if I want to select all the ones whose income is greater than, say, 20,000. Right? So income is greater than 20,000. Add the condition. I could add another condition based on another column. I can add as many conditions as I want. When I have multiple conditions, I can then basically AND them or OR them. AND means they must all be true. OR means any one is true. So I can, at the moment, it says all conditions are met because I only have one condition. But if, for example, I said log income is greater than 5, this is not a very sensible one to add in, of course, because those are basically overlapping. But you see, I can choose either if all conditions are met or if any condition is met. Right? And then I can choose OK. And you'll see, in fact, I made it a very broad condition. So now lots of my rows have been selected. And if you come back here, you'll see, I can see many of my rows have been selected in here. So that wasn't a very suitable selection. Let's try again. Let's just choose the one I originally chose, log income. Well, let's just choose income was greater than, what did I say, 50,000? Let's say 100,000. Add that condition, okay. So you'll see that's still quite a few. And then again, I can do something else. I can put all of these to be maybe be red. So that's quite a large number of these are shown up. But if I now show the distribution instead for log income. You'll see it's only the extreme values on the log income scale. Right? So it was a problem on the income scale, it appeared to be, but actually that's because the distribution looks very different. So log income indeed is a symmetrical distribution, as it should be, because I generated the sample from a normal distribution. So this does indeed look like a normal distribution. Whereas on the income scale, obviously, it's a very skewed distribution. Okay, so that's how I can select. What haven't I done? I can also do it manually. Oh, I can even hide those rows. So if I hide those rows, then they will be hidden from my graph. Right? So they've now disappeared off there if I redo this. So let's now do my distribution again. So you'll see that the high income ones have now disappeared because I've hidden them. Right? So I can just temporarily hide some data values. This way, you'll see it's highlighted, it's put these sort of mask over the eyes, right? It's put a blind over the eyes, meaning these ones will not be shown. I haven't deleted them from the data file, but they're hidden from my analysis. We can also exclude them. So hide means they don't appear on the graphs and they don't contribute to calculations. Exclude removes them from the calculation. So let's turn, and these are both, and hide, oh, sorry, hide and exclude means I don't show them and I don't include them. Hide alone means just I don't show them. They're still included in the calculations, right? So I have three different options. And then these are all toggles, meaning if I keep them selected, if I click on hide again, it will unhide them. So this is going to be very useful. So later on, when we want to examine the consequence of potentially removing some data values from our analysis to see whether it changes our conclusions, I can use exclude. So for example, if I now do exclude, you'll see it now puts a different symbol against these rows. It puts a stop sign, meaning these will not be included in my calculation. If I now look at the income distribution, right, you can see it stops now at 100,000 because that's where I made the cutoff. Okay, so I've removed the most extreme incomes from my distribution by doing this, and similarly, but here they're actually excluded from all my calculations. So those are very useful tools for examining sensitivity of my analysis. I don't actually have to delete them. Right? Generally, we don't want to delete things, otherwise we may forget what we've done. But this lets us try with and without the data file, the data points, and we can also show and not show them using hide and exclude. Okay, so that's very helpful. What haven't I done? Right, so, oh, and then once I've got my results, how can I use them? So let's say I've got this analysis here. How can I get access to this? So again, if we go to the three dots, you will see that we can export this output, right? So we can export this output as text. Obviously, that means we would lose the graphics. We generally won't want to choose that. An image means it's all one big image. You can choose different formats. Generally, we're going to want to save it as something like Word. If we save this in Word, that means, you see, it's creating a file named distribution.doc, means that this distribution analysis will be in a Word file ready for us to use in our report. Okay? So that's very helpful, that you can save all of this analysis in the report. The other thing we can do in order to remember what we've done is to save the session script. So the session script means a record of what analysis we've done. So we can rerun the analysis later. Right? So this is a very helpful way for you to keep track of what analysis you've done, so you can document your analysis. Okay, so the session script is also extremely useful. Okay, so these are, we can also do PDF, RTF. RTF generally is not very useful anymore. So these days, even if you don't want to use Word, if you want to use the Google Docs, for example, I think Google Docs can read Word files, right? So these days, it used to be that because the Word document format was not open, that people would use RTF. RTF is an exchangeable version of Word, if you like, but you lose some of the information. So generally, I wouldn't recommend that anymore. The other thing, what other options are important here? Let's have a look. Yeah, you can also export the data set itself. So from the data table, usually the most convenient form is to use Excel. Because most statistical packages or other forms of analysis can read the Excel file. So if it can't read JMP, usually can read Excel. In fact, SAS data sets are also very often read by other statistical packages. And that way it contains more information than is contained in an Excel workbook. Okay. Now, when it comes to the report, I do not require you to use JMP. You can use R or SPSS or Stata or whatever else you would choose to use. But to be honest, unless for some reason you don't want to use JMP, JMP is generally much easier as a tool. Okay, I think that's a good time to take a break. We've already been going for an hour and a half. Just give me a few minutes to have a drink of water. So I'm not going to stop the recording. It's easier just to keep it going. Let's see, how are we doing now? Do we have more people attending? Where's my... Right, so Edmund has joined us. What about Rajesh? Has Rajesh joined us? No. I think everybody except for Rajesh is either here in Zoom or in the classroom. Okay, so for Edmund, you missed the beginning. So I'm recording the session in Zoom, so you'll be able to watch back what we've done. And I asked at the beginning to check that everybody has successfully downloaded the JMP trial. If anybody has a problem, you can communicate with me in chat. Okay, so you don't have to communicate openly in chat. You can send me a direct message if you prefer. Okay. So the clock here says 11:10. My watch says 7 minutes past. So let's start again maybe at 11:15. We can finish a little bit earlier. Okay? Okay, shall we start again? Everybody ready? So we talked about row selection. We talked about exporting data. What about manipulating tables? So this can be very helpful. So sometimes we need to reorganize our data table. So there are many different things we can do. So for example, we can sort the records. So if I wanted to sort by the log income in my table, so I could easily find different information, then just doing that. Now my data is all sorted, so I can easily see the low income and the high income ones. It's already sorted in that format. So you can see it goes from 24.6 all the way up to what? 6 million. Yeah, so that's obviously too much variation in my example, right, to be realistic for income. So what else can we do? We can do a subset. So for example, the obvious one would be I could select the rows. So I could have done the row selection in any way I like, and then just those rows I've selected become the subset. Or I can even do a random sample. And I can do random sample in two basic ways. I just say what fraction I want, or I can say what sample size I want. So for example, at the moment I have 500 records. I could say, actually, I only want 100 records, or perhaps for the report, I want just 50. So if I say 50, I now have a sample, which should be a random sample of 50 from that original 500. So there are different things I can do like that. Let's get rid of these. I don't want to keep that. Get rid of this one as well. What else can I do? I can do a transpose. So transpose is not usually useful. Transpose means swapping rows and columns. So for example, this would be very unhelpful for this data set because that would give me 500 columns and 2 rows. Right? So there are very few situations where that's useful, but sometimes it is. And then stack and split, I will illustrate in a moment. Join means I'm joining rows by matching value. So if I have 2 data sets on the same set of individuals, and I want to match them together on that row identifier, I can do that way. Concatenate means I'm adding rows from different sources. So that might be I have 5 different samples, all of which have the same variables. I could add those together in one big file. So those are all very straightforward. So the stack and split is a little bit more tricky. So we need to look at an example. Let's see if we can find that sample in here. So we're going to go back to I'm going to go out of the import data, back up a level, the samples and data. Now they've changed the name of this one. Let's see if I can find it. Yes, the cheese tasting one. One of these is in a different format. I think it's the one called cheese taste I want. Yeah. So this cheese tasting one, the data is in a summarized form. So I need to explain what this data actually means. In fact, I think, does it say in the notes? I think it does. But we need to I want to see the notes here. Yeah, so this data set is testing on cheese, right? So you ask people to taste the cheese and you ask them to rate it on a scale of 1 to 9. Okay? So A, B, C, D are our 4 cheeses and 1 to 9 are the ratings. So what we know is that, for example, cheese A, nobody rated it 1 or 2. One person rated it a 3. And then the most popular rating of 7 was from 19 people. So if, for example, we look at the mode, you can see D was rated the highest with a mode of 8. A was next highest with a mode of 7, and so on. And what's the worst? B is the worst, right? The mode was 3. So in other words, each row here is not really a row of data, right? What we want to do is to do the stacking. In other words, we basically want to know, we want to have as a data set that says for each cheese, how many people rated it at each level. So this is a summary of the data, but it's not data that we can easily analyze as it stands because we cannot compare different columns on the same row. So, see if I can get this right. I often get this wrong. So stack. We basically want to stack on top of them. So we want to stack A, B, C, D and keep the response. If I've got it right, let's see if I've got it right. So that means that we now are showing that each combination of response and cheese. So this one that's called label is actually cheese label. I can't spell cheese. And the response is actually a rating. And the data is actually the count. Okay, so this tells us 0 people rated cheese A as 1. Okay, so rating is an ordinal scale assessment. This is telling us which cheese it is, and this is the count. So this data set is now something we could use. And of course, we could regenerate that table from this. But this would enable us to do an analysis. Okay? So this is now structured. So sometimes somebody has created a summary table and we need to restructure that and essentially split. Let's just do the reverse. Right? So this is just the reverse of stacking. Okay, so those are not things that you're going to use very often, but they are there for those situations where you need to do it. That's obviously a lot easier than trying to do it manually. Okay. What other things do we want to do? Yeah, I don't want to talk about transpose. I've shown you sort and subsets. Okay. Now the formulas. So the formulas is really very nice in JMP. So what I love about the formulas is, so in Excel, we have formulas. And in fact, these days in Excel, you can create a formula on a column. But traditionally, people put formulas in cells in Excel. The problem with that is that sometimes the formula modifies itself in ways that you don't really understand. So there are classic examples of people who've used Excel in ways where they don't know quite what Excel is doing. I don't know if you followed, even for vaccination in the UK, sorry, not vaccination, for the virus, for COVID. The UK actually had a major problem where they had failed to follow up on some people who had the virus after testing because they were using an ancient version of Excel, which has a limit on how many rows. So any rows that were beyond the limit, they lost. Right? So it's quite easy to get things wrong in Excel, particularly the most common reason is because people have copied a formula across cells and not correctly understood how the formula varied across the cells. So the beauty of JMP is it's more disciplined than that. It forces us to create a formula at the column level. And that means that just like that example I showed you with the income, if I change, in fact, if I change the log income, the income column will automatically update to match. Right, so we'll automatically do those adaptions. So let's have a look about what sorts of things we can do. So some of these can be quite useful. So let's see if I can find the correct example. So there's one called students. If I can find a students one. We've got more students. Let's try students without a number, see if that's the right one. Yeah, I think this is the correct one. So this is the height and weight and gender or sex of a group of students and their age. So the age varies. Whoops, sorry, the age varies. I've lost my window. Just a minute. So we don't need to worry about the ID number. We're not going to use that. That would only be useful if we wanted to match it with another data set. So the key thing here is the height and weight. So this is the absolute height and weight. But what if, for example, we weren't really interested in their height and weight, but how their height and weight varied relative to everyone else? So we know we might want to standardize it. So how might we do that? So we can standardize the height and weight by creating a new column, which is the standardized version of this. So we can actually do this in different ways. Let me just standardize height first. And I'll put STD in brackets to remind me that this is supposed to be the standardized one. And then I'm going to put my formula, so I can either right-click on the height to get formula, or I can, if I don't remember, I can go to columns after selecting it and choose formula there. So what do I want the formula to be? Now, the easiest way to think about this is, okay, so I want to start with the height. So to standardize it, I want to subtract. What do I want to subtract? I want to subtract the mean of the height. So I select height again, and then under statistical, I can choose the mean. Now be careful, because there are two different means here. This says returns the sample mean across rows in a column, whereas if I choose the one down here, chooses the arithmetic mean of the values within the argument. So for example, if I chose this mean here, I get 0, because it's choosing the mean of that particular value. So that's the wrong formula. What I need to be careful is to make sure that what I'm putting in here is the column mean, not the statistic, not the straight arithmetic mean. So how do I get rid of that? So up here in the editing tools, you will see there is an insert and a delete. So you may not think about this. What does this symbol mean? So this is when you edit a document. So these days, probably all of you write your own documents in Word or something like that. But traditionally, I still remember when I joined the statistics department, we had typists who did typing of all our papers for us. So they would type up the paper, and there were always mistakes. So you had to mark on the script telling them what to change. So you would put this symbol for inserting something. This symbol means delete something. So I click on this, it deletes that. Again, I'm going to go back and start with height. I go to my statistical formulas. I want the column mean of height. So if I now choose apply this, you will see now the heights are standardized in the sense I've subtracted off the mean height. But actually, I usually want to turn it into what's called a Z value, which means after subtracting the mean, I divide by the standard deviation. Okay, so I can now click on the divide option here, and then I can put underneath it height, statistical, column standard deviation. Apply this. Click OK. Now let's check whether I've done it correctly. If I've done this correctly, I look at the distribution of this new variable. I should have a mean of close to 0 and a standard deviation of close to 1. Not a standard error. Remember, this is a standard deviation. In fact, it says the mean is 6 times 10 to the minus 16, basically 0, right? And the standard deviation is 1. So I've done the standardization. So then this basically has turned it into a z-value, right? So this is telling you the height of this particular child compared to all the other children. We've taken off the average height, so we know relatively how tall this child is compared to all of our other 233 children. Okay? So that sort of thing is quite useful to do. Now, we can do other things as well. Now, for example, we might want to do recoding. So let's have a look at this. So we're going to try the hot dogs one. So I went to a burger shop yesterday and it reminded me how horrible burgers and hot dogs can be in Hong Kong. Okay, hot dogs. So the hot dogs one, in fact, if I remember correctly, only these, no, it's not a font, isn't. Yeah, this has got value labels. So we don't have any formulas in here to start with. So for example, what might we want to do? So we've got here 54 different types of hot dog. This is obviously US data. For each type of hot dog, we've got what type of hot dog it is. Now, is it beef? Is it chicken? Is it meat? Meat basically means they haven't told you what's in it. Oh, sorry, poultry is what they call chicken or duck, I think. So we've got, so poultry or beef is not too bad. We basically know what it is. Meat means it could be anything in there. I mean, it's illegal to put horse or dog probably, but in the past, people would use horse meat because it was basically cheap and horrible. We've then got the taste, and we've got the price per ounce. We've got the price per pound of protein. I mean, this is America. They still don't live with the metric system. We've got the calories and we've got the sodium. I don't remember quite what the protein fat issue is here. Anyway, let's say we want to recode it based on the calories and the sodium. So let's look at the distribution of calories and sodium. So I've selected both these columns. How did I do that? I selected one, I held down the Shift key and clicked on the second one, right? Just the same as any other selection operation. Analyze distribution, select both of those. So this tells me that the median calories is about 150 and the median sodium is about 400. So I could, for example, divide up all the hot dogs into 4 groups based on whether they are low or high on calories and low and high on sodium, right? So how would I do that? So I'll keep this here to remind me to make sure I can try and get this correct. So I'm going to create a new column. So I'm going to call this what cal sod to be, because this is about calories and the sodium. And I want to create this as character because I want basically the answer to be text. It will be nominal scale. And then I'm going to put a formula in here. See if I can get the formula right. So So first of all, I said, let me look at the low calorie, low sodium. So I'm going to create that one first. So how do I do that? So I do conditional. So conditional, I'm going to start out with an if statement. And you'll see it gives us a very simple if statement. So if X, then Y. If not X, implicitly by meaning else, then something else. So my expression here will be the calories are low and the sodium is low. So how do I do that? It's a comparison. So amongst the comparisons, I can say A less than B. So that means if this will be calories is less than, so I've said the median was, I can see it was about 150. It's 145, but I'm going to keep it simple. So if the calories is less than 150, and then here, now remember this, it's going to be, I want it to be text. So I want to put in quotes LC. In fact, maybe I'll just do calories for now. We can do this in a minute for calories and sodium. So if it's not low calories, then I want to put it as high calories. Okay, have I got it right? I click OK, it's fine. Now, if you had put in that calories and sodium was meant to be numbers, then you might have got an error message, right? So now, but let's say I really want to do it properly. I want to do all 4 categories. So I don't just want to do this. So how would I do this? So if I click on calories less than 150 and I click on conditional again, what happens there? So now I've got a 2-level if then else, right? So if calories is less than 150, then that's still not quite what I want, is it? That's not what I want. So let's not do that. Let's take that out. What do I really want in here? If I click on the else here and put conditional, that's closer to what I want, right? I basically want if something else, if something else. And then similarly on this one, I also want a conditional if. Right? So if, so first of all, I'm going to separate it by whether the calories are high or not. Then I will separate it by whether the sodium is high or not. Okay, so if calories comparison, let's do less than rather than greater, right? So if calories are less than 150, if not, so these ones are all high calories, these are all low calories, and then here, if also the sodium is low, and I said that the median was around 400. So if the calories are less than 150 and the sodium is less than 400, then I'm going to call this one LC. I'll edit this to be LCLS. Okay. So that's the low calorie, low sodium group. Otherwise, it's low calories, high sodium, right? So I will put this one, low calories, high sodium. And then in here, I can copy this. I can do copy and paste. Although I can't remember how to do copy on this keyboard. See if I've got it right. Yeah, copy and paste. So this will now be high calories, low sodium. I'm double clicking on here to get to here. And then this one means it wasn't low calories or, sorry, it was high calories, high sodium. Got to put it in quotes because it's text. Got it wrong. Let's try again. Oh, I've got a double quote. So if the calories are low and the sodium is low, it's low calories, low sodium. If the calories are low and the sodium's high, it's low calories, high sodium. Similarly, right? If the calories are high and the sodium's low. So if I've done it correctly, I should now have my 4 groups. I can go and check whether I've done this correctly. So let's have a look. How can I easily check it? I can do fit y by x, where I'm going to look at the calories and the sodium together as my y, and my x is my new coded variable. Right, so that should show me. So how high calories, high sodium. So for calories, high calories. Oh, I get something wrong. Oh, yeah. So if I've got high calories, low calories. Oh, I typed something wrong, did I? Has it got my formula wrong? Let's check my formula again. That's very odd. Calories and sodium against my new variable. Oh, I see. That's just showing me the variable name. So you can see the high calories ones are high for the 2 HCs, and the sodium is high for the 2 HSs. Okay. So what does that mean? It means I, for example, could look at the price relative to the calorie and sodium context. So I could look, for example, whether the price per ounce is related to the calories and sodium. So this is just giving me the graph, but I can ask it to do the analysis of variance. So this is now showing me both the graphic and the analysis of variance. So it's showing me that if we look at this down below here, you will see low sodium, high calorie, low sodium is more expensive. That's the main difference, right? I think. Let's have a look. Yeah, the high calories, low sodium is the most expensive, followed by low calories, low sodium, followed by high calories, high sodium, and the cheapest are low calories, high sodium, right? So it's not a very clear pattern. It enables us to do all these different forms of analysis by creating these new coded variables. So it's not hard to create it. And the beauty is, if I'm not sure, I can always come back and check my formula to make sure I've done it correctly. And if I discovered a mistake, I can fix it. If I decided that actually that's the wrong cutoff, I want to make the calories cutoff at 200, I can just change it here by double clicking on this, typing 200, applying that. And now I've just changed the definition very easily without having to go back and fail to remember what I've done. Okay, so this is the beauty, is you've got documented implicitly how you've created these variables. Okay, and it's consistent across. You don't, if you're worried about whether the formula is correct, you just have one place to go and check. Okay, what else?
Sorry, Professor, can you hear
Yes.
me? How do you do the analysis of variance from the graph?
It's not from the graph, it's from the, so let me do that again.
Thank you.
We're going to look at that more carefully later, don't worry. This was just to illustrate what I've done. So I looked at the dollar per ounce against the calorie sodium breakdown. So in the initial stage, JMP just shows you the graph, right? Hey, what's happened to the rest of my data? Something's gone wrong. Oh, because I've made it so now there are no high calorie ones. Let me just change this back. I changed this too far. Okay, so 200 calories is too high a cutoff. Let's change it back to 140. Yeah, so we're going to talk about analysis of variance more carefully later on, right? This was just to illustrate very quickly what's going on. But when we do the fit y by x, JMP initially shows the graphic display, right? So we've got the 4 possible values for the cal sod variable, and we've got dollars per ounce here. But then if you click on these little click down arrows, you can choose which analysis to do. Okay? So we're going to go through this much more carefully later on. So I just chose quickly means analysis of variance. Okay?
Thank you very much.
We're going to look through these much more carefully. And when I choose that, in addition to the formal analysis of variance, it also shows me adds the diamonds in here, which is basically showing you the means and standard deviations as well, right? So you can get a better feel for what's going on. Okay? So anyway, don't worry. We're going to come back to analysis of variance and all the analysis later. I just wanted to illustrate how the coding has worked. Yeah, so the other thing, so the transformation I've already illustrated with you. I showed you on the income one how we could do log and inverse log. And we've already done the random example. Okay. So the data display, we've already looked at a few of these things, but not others. So let's start with univariate distributions first. We already looked at a couple of bivariate ones. So what are the key questions? Why do we do a univariate display? So the sorts of questions we generally are asking, at least implicitly, are whether the data is unimodal. Why do we care about that? Well, if there's more than one mode in the distribution, then maybe there's a more complex process generating the data, right? Maybe we can already see that there might be 2 processes generating the data. The other question we often want to ask is whether it follows at least approximately a normal distribution, possibly after transformation. So we will look at some examples of this. Now, how do we test for normality? So usually, I don't encourage using formal tests of normality, but rather we use graphical displays. So we're going to use something called a normal quantile plot in order to illustrate. So normal quantile plot basically means we compare what distribution actually is compared to what it should look like if it follows a normal distribution. So in other words, we should get a diagonal plot if the data really comes from a normal distribution. Okay? So that's very useful to help us understand how it may or may not be. So if the data is not normal, what we can often do is some form of a power transformation to turn it into something that is normal. Okay? Now, I don't want to go through all the text here. What I want us to do is to go and start looking at the example data sets. So let me just come back to that. So we're going to start to work our way through the data sets in the course book, in the DA course book. So we're going to start with the data file that's called diet. Okay. So what is this data set? This data set is not to be taken too seriously, but this data set is supposedly showing you the change in weight of people after trying two different dietary examples. Now you'll notice, although there are 500 rows, that for the Quicks weight change, many of the rows show missing values. That's because these are really two separate data sets. Right? We've got the data from Quack's weight change, and we've got the data from Quick's weight change. So let's have a look at the distribution for these two variables first. Okay. So what does JMP show us? So again, initially, the focus is on purely the graphics. So we can see side by side the distribution of the data. So for Quack's weight change, we have 500 values, and it looks pretty symmetrical, right? It looks pretty much like a normal distribution. For Quick's weight change, it doesn't look very much like a normal distribution. But remember, there are only 20 data points here. So it's much harder to see what the distribution looks like when you have so few values. But we can actually check whether it's approximately a normal distribution. So one of the options here is a normal quantile plot. So let's just show this so you get some idea how this applies. So this normal quantile plot, as I said, what it's actually showing us is comparing what the data looks like compared to what it should look like if it follows a normal distribution. So you can see our reference is really the diagonal line. So the red diagonal line is what we expect the data to follow roughly. And then the two curves are essentially our statistical test. So if our data points are within the curve, then we don't think it's non-normal, right? So you can see actually, with the exception of these data points right down here and up here, this is pretty close to a normal distribution. So there's no real reason to think that this is not a normal distribution. But in fact, if all we care about is the mean value, we know that the sample mean for a sample of 500, the distribution of the sample mean certainly follows a normal distribution. So the fact that the data doesn't follow a normal distribution isn't that important if all we're asking is, what is the mean? Okay. Now, that's not going to be the case when we come to some of the models later on. In those cases, whether it follows a normal distribution actually does matter. But in here, all we're basically saying is, does this diet work? So the real question is the mean 0 or not? Okay. So in this case, you can see the mean is minus 0.9. The standard error of the mean is 0.44. And the 95% confidence interval goes from minus 0.05 up to down to minus 1.77. Right? In other words, we can implicitly have got a test of the mean. We can actually ask for a test of the mean. The hypothesized mean is 0. We don't know what the standard deviation is. We're relying on the data, so we can leave that blank. We can also ask for a non-parametric test, just in case we're worried about normality. As I said, the non-parametric matter shouldn't matter very much in this case because we've got a relatively large sample. So let's have a look to see what it says. This tells us what? So this down here is telling us the statistical test. Now you have to understand the jargon that JMP uses. So we're given 3 different observed significance levels here. So the first one, what does this mean? Probability greater than bar T bar. That's just shorthand for saying absolute value of t, meaning this is a two-tailed test. So that's just JMP's way of writing a two-tailed test. So we don't care whether the deviation is positive or negative. Actually, arguably, we do care, right? All we really want to know, what we really want to know is a one-tailed test here, arguably. We're not interested in whether the weight is 0 or a little bit higher. What we really care is there any evidence that it decreases the weight? So if we're only interested in that side, then arguably we just look at the one-tailed test, right? Because we should have been saying in advance, we're interested, is there any evidence that these people have lost weight? So this says the t-test, which is the one based on the normal distribution, the one-tailed t-test is significant at 1.8% if we use the one-tailed test. And the signed rank test, which is the non-parametric one, is significant at 2.7%. So it doesn't make a big difference whether we assume normality or not, right? Because we've got a big enough sample. Essentially, the sample mean follows approximately a normal distribution anyway. Now, we have to be careful here because this is simply saying, is there any evidence that on average, people lose weight during this intervention? It says nothing about whether the change is meaningful. So if you look at it, this is US data. This says these people on average lose less than 1 pound. So do you really want to do all this effort just for the sake of 1 pound? Right? Your weight varies by several pounds depending on how big a meal you've had today. Right? So this is basically, yes, there is evidence this works, but the change is trivial. Right? It's too small to be meaningful. Okay, let's compare that to the other one. Okay, so let's compare that to Quick's weight change. So what can we say about this one? So this one, the mean is minus 2.73. So that's actually still not a big change, but at least it's nearly 3 pounds. It's more than 2 pounds. But, well, also the standard deviation is similar. The standard deviation for Quack's weight change was 10 roughly. This is roughly 11. However, we only have a sample of 20. So the standard error is much bigger because we simply don't have a big sample. So you can see the 95% confidence interval goes from roughly -8 up to 2.5. In other words, clearly we cannot reject the null hypothesis. So let's do that again, formally in just the same way. So this is telling us the one-tailed t-test, 14%. And in fact, the one-tailed non-parametric is about 7%. So in this case, we have a relatively small sample. So with a sample of just 10, the normality assumption becomes more important. But ironically, the non-parametric test says there's stronger evidence than the parametric one. Right? But the key issue here is that we need to be careful. This is basically saying it's not that this one is less effective than Quack's. It's simply we don't have enough evidence. Now, it could be that if we did a sample of 500, we would find out this one isn't useful at all, right? But at least the average change is better for this intervention than for the other one. So we need to be careful whether this is meaningful and significant in the big sense of the word significance is quite different from whether it's statistically significant. So that's one of the reasons why people criticize the language of statistical significance, because people interpret it to mean important, whereas actually all it means is there any evidence as to whether it's non-zero? Can we reject the possibility of it being 0? Okay, so quacks, we're fairly sure it's not 0, but its true value, right, is somewhere in the range. Where was our confidence interval? Somewhere in the range of minus 0.06 up to down to minus 1.8. Right, so that means almost certainly it doesn't reduce your weight by more than 2 pounds. Right, it's somewhere in the range of minus 0.8, means less than 1/10 of a pound up to 2 pounds. Whereas for this one, we're saying it's somewhere in the range of losing 8 pounds to gaining 2 pounds, 2.5 pounds. So we simply don't have enough statistical evidence. So we didn't look at the quantile plot. Let's look at the quantile plot of this one. So this one, the values are not as close to the diagonal apparently, but again, that's largely because we have a much smaller sample. You will see actually the other one arguably goes just as close to the bounds as this one does. Right, so although this one doesn't look like a normal distribution when you just look at the histogram, the normal quantile plot is saying you really don't have any evidence to reject normality here. So again, doing the non-parametric test does give us some assurance that our conclusions do not depend too much on whether it's a normal distribution or not. So it's a good idea to have done the non-parametric just to confirm, right? At least we have some reassurance that. So as I said, if you were writing your report and this was what it was about, you could say, you might report the t-test and say the t-test shows that it's not significant at 5%, but the sample size is perhaps a bit small. But you could say we also did a non-parametric test and the significance level was no greater, right? It was actually slightly smaller, but it's still not significant at 5%. So we know that the conclusion does not depend very much on the normality assumption here. Okay? So that's our first data set. How are we doing? Let's just do the second one before we finish for today. So the second one is Gosset's corn. Does anybody know who Gosset was? Anybody have any idea who Gosset was? Gosset actually should be famous. Why should he be famous? Because the student in students' t-test, so think about it, we talk about students' t-test. Why is it called students' t-test? Because the person who wrote the paper called himself student. His name was not student, his name was Gosset. And this is the data set he used in his paper. So why did he call himself student? The reason why was because he worked for a company. He was not an academic. In fact, the company he worked for was Guinness, right? Guinness, the company that makes stout, that dark beer, which you can buy. So he worked for Guinness. And the thing he worked on was about the statistical control of the process for making Guinness. So one of the key things when you're making beer or stout and similar things is that you often have to dry the grain before starting the fermenting and brewing process. So this data set is comparing two different ways to prepare. Now, I never remember in Guinness. So beer is normally hops and barley. No, hops and I can't remember. Anyway, I think this is barley, if I remember correctly. I can never remember which grain they use in Guinness. So this is comparing the results from two different ways of doing something, right? So actually, this example is a paired example. In other words, what he did was when the barley or the grain came in, he would divide the grain up into two parts. One was done through the regular process. The other was dried in a kiln. So a kiln is basically a big clay oven. So we normally in English use the word kiln when we're making pottery, right? So after you have made your cup or your bowl out of clay, you put it in a kiln to bake it, right? The baking process to make it so that your cup will not fall apart, okay? So a kiln in this context is a big drying oven. So they were comparing the standard way of drying the grain with this new kiln to see whether the kiln gave better results. So that means we actually have to create a new variable, difference, right? So we can create a new difference variable. So we just do kiln minus regular. Apply. Okay, so we now have our data to analyze. So we can look at the distribution of those differences. We can, if we like, do the quantile plot. Quantile plot looks fine, right? They're not that close to diagonal, but this is a small data set. So the key thing was that what Gossett did in that paper was to say, if the sample is small, right, how do we handle this? Because that means because we don't know the variance, then that test, we're estimating the variability, the variance as well as the mean. So we need to take this into account when we do the test. So we need to do the t-test rather than doing a z-test. So if we already knew what the standard deviation was, we could use the z-test, but we don't. And in these sorts of paired situations, we almost never will know the variability of the differences. Right? So if we do our test, and again, Wilcox and sign rank test didn't exist in those days. We can compare the results. So this tells us the two-tailed test is not significant at 12%. And interestingly, the non-parametric test gives you almost exactly the same result. Right? The significance level is also 12% for the non-parametric test. Right? So we do not have enough evidence to reject the null hypothesis. In fact, the mean is 33, the standard deviation is 66, the standard error is about 20. Right? So again, this says if you wanted to check whether this might be significant, so if you really thought the difference might be around 30, you would probably need you would need at least double the sample size, probably, in order to be able to detect it. But of course, the question could be not just statistical significance, but is a difference of 30 in the outcome important or not? If it's not important in practical terms, then why would we care? So this is probably the yield from the brewing process, right? So which way round have I got this? I think this is kiln versus regular. So this seems to suggest that the yield is slightly greater for the kiln over the regular process. So if 30 units increased yield was important, maybe Gossett should go back and collect more data, right? But if 30 units is not even close to being important enough to change your process, then this is saying it's probably not worth it. Okay, so again, we need to distinguish between statistical significance and practical significance. Okay, so this is just a very famous data set because this is the one that Gossett used in his paper. In fact, I think he wrote it with Pearson, which led to us talking about the student's t-test. Okay. So I think that's a good point to stop. Next week, we're going to move on to looking at some other situations. We're going to look at transformations of data, and we're going to come on to analysis of variance. We're going to look at the difference. In fact, we're going to look at the difference of boys and girls on height and weight. And we're going to then start to move on to linear models and general linear models. Okay? So I'm sorry we had a bumpy start today. For some reason, my laptop using the Apple adapter will not work with the HDMI cable in here. So I will bring in a different adapter next week. If that fails, then we'll fall back on what we've done today, which is to use Zoom on the lab computer. So what I want you to do as your homework this week is to start looking for the data set. So this is the data set for your analysis. So at the beginning, I took you through what the requirement is for the report. So one of the things in there it says is that I need to agree that your data set is suitable. So generally, the issue is if your dependent variable is not continuous, then you need to persuade me that it's still a reasonable data set to use. Okay? So you should start thinking about a data set which is meaningful to you and meets the requirements for this course. You are then welcome to email me the data set if it's not too big. If it's very big, you can send me a small sample of it plus an explanation of what it is. So probably as a JMP or an Excel or an SPSS file. So I can sign off on it so you can start analyzing that data set as we go along. Okay? So the sooner you find a suitable data set, the better, because then you can start to do things as we follow along. Okay, any questions? Oh, for the reports appendix. So somebody asked whether I prefer the output to be a picture or as a table. So that really depends on the context. So obviously, if you're talking about you had done the check on whether it's a normal distribution or not. So you're using a normal quantile plot, then obviously I want to see the plots. I don't need a table. On the other hand, if you're talking about how well different models fit, I would normally expect to see a table comparing those models on the criteria, whether it's adjusted R squared or AIC or BIC. Right? So it really depends on the context. But I don't expect a lot of output. I just expect the minimum amount of output in order to confirm the results which you refer to in the report. Okay? So we can talk about it a little bit more when we're a little bit further along. Okay, at this stage, what I want you to focus on is finding a suitable data set to use for your report. So it can have a lot more cases than I've mentioned. I don't mind if it has 2 or 3 or even 5,000 cases, because that generally doesn't make a big difference. That's really just a minimum. And similarly with the number of variables, I've said I want at least one factor amongst independent variables and at least one covariate. Can I have more than that? That's not a problem. So those are really minimal requirements for the data set for your report. Okay? Any other questions?
Professor, I do have another question.
Yes.
So in your past iterations of this course, has anyone proposed to do data set analysis on cellular RNA sequencing data, like big data kind of?
No. So the main reason, I think, is because that raises somewhat different questions about having a very large number of variables, right? And also because generally that's doesn't illustrate very well the things that we're talking about in this course. So I won't say it can't be done, but so in other words, large N, large samples is not a problem. A very large number of variables may be a problem. In a sense, it doesn't illustrate very well what we're going to be talking about in this course. Right? But if you want to try and persuade me, that's fine. You can send me you can email me explaining why you think that fits within the context of this course. Okay?
Okay, thank you.
So, I mean, did you attend my big data workshop this last week?
No, I didn't.
Yeah, so I did talk a little bit in there about large P, meaning basically large number of variables. So some of the issues can be addressed within the context of this course, but I don't think it's a very good way to generally illustrate linear and generalized linear models. Right? It's generally, most of the problems are essentially dealing with very large scale analysis of variance, where you're essentially looking at a very large number of different genes or something similar to that, correct? Is that the type of problem you're talking about?
Yeah, so me looking at gene analysis, but I think it's too ambitious, so it's okay.
I think just generally it doesn't fit very well with what I'm trying to teach you. So there is an issue about a section about false discovery rate in the analysis of variance chapter, right? So there is some relevant material, but JMP is not the sort of tool I would be recommending for that.
Okay.
There are also some additional issues around scaling. So I'm going to talk about compositional data a little bit later on. So often the genomics data is really what I would call compositional data, in a sense that there is an unknown scale for a lot of the output, and people often ignore that. So there are some specific issues around genomics data which generally need special consideration. Okay.
Okay, thank you.
But again, as I said, I'm always open to a discussion if you want to try and persuade me that your favorite data set is a suitable one. So I've said, for example, that the dependent variable should be continuous. If the dependent variable is count information, then that probably can still be looked at. The issue is that the diagnostic tools are somewhat different, right? Rather than the ones we're going to primarily focus on. But it's enough within scope that I'm willing to consider that. Okay, any other questions? If not, thank you all very much. So I will put the Zoom recording. So I will not share the recording directly in Zoom. What I generally do will be I will take the Zoom recording and put it in the Google Drive folder and then send you all a message when it's there. So Zoom these days is working pretty efficiently. So usually within today, they will have sent me the link to the recording. So hopefully by tomorrow, I'll be sending out a message that I put the recording into Google Drive. I'll also put it in a YouTube folder so that you can access it via YouTube instead. You don't have to go and download it from Google Drive. Okay? If you have any problems accessing the materials, please let me know. As I've said, you can presumably get the 2 PDFs already, and I will be putting the additional data sets in a zip file in the Google Drive folder later this week. Okay? But for at least all of next week, we will still certainly be using the sample data files inside JMP. So you shouldn't need to access those yet. Okay? Thank you very much. Any problems from you guys?
No problems. Just want to say thank you because your introduction to the software was very clear.
Okay, good.
Thank you. Bye.
Bye-bye.
Thank you. Thank you.